Hi, this is Francis Christian for general surgery in quarantine. That is a term that I borrowed from my son who has a YouTube live series for his students. It's called Economics in Quarantine. Can you indicate to me whether you can see and hear me? And then we shall start. Once I know that you can see and hear me, uh, we'll start. Oh, hi, Annalise. Good. I'll just wait for maybe a minute, and then once we know how many people are in this group, we'll start. Okay, so this is economics, not economics, but general surgery in quarantine, because there is an economics in quarantine uh, YouTube live series that my son started for his students, and you can find that. So I just want to point that out. Yeah, and then we will start our general surgery in quarantine second YouTube live seminar. Today is a topic which I plan in two parts. I think it's better, in fact, to put it in three parts. And at the end of today's YouTube live seminar, what I'd like you to do is to indicate to me either by text, email, or some other way whether you think I should condense two and three parts into the second part, or whether the the whole uh, topic is exciting and arresting enough for you to have this in three parts, which I think is better. But you let me know. Now, you'll realize that the slides in my seminar today are a little bit more detailed than usual. And that is because I'd like it to be a ready reference for you later on as well. I'd like it, for example, to be something that you'll go back to when you don't know the meaning of a particular word. Molecular biology is full of different terms and terminology, and you can get lost in it if you don't understand the principles. So today, what I'd like to do for you is to enunciate those principles. And then if you remember those principles, you can always come back to my lecture, and you can then know which uh, terms mean what, and everything will make sense to you. The other thing, of course, is that you'll notice two or three typos in my slides today. I'll correct those when I send the slides out to the SlideShare uh, website, as well as when I send it out by email. Those are minor typos. What I'd like you to bear in mind as we go through this seminar is the fact that there is in all my slides the attempt to make new terms visible for you on the screen as soon as you see them. In other words, every time you see a new term, I've put it in capitals. So any term that you need to define, either now or for later, you can always go back to and you'll find it there in capitals. So when you see capitals, it means I'm introducing a new term or a new concept. Okay. Now, let's look at why the surgeon must know molecular biology. And in this slide, I've also put on the right side of the slide what the 4 essential areas of molecular biology are for the surgeon or for anybody else. Now, you would know that there are multiple areas of therapeutics which the surgeon needs to be familiar with, or the synthetic antibodies that are now being made, all these molecular targeted therapies, excuse me, which are now a huge thing in cancer, but also in other areas like hepatitis C. So molecular biology is directly related to multiple areas of therapeutics. It is also basic to multiple areas of diagnostics. With the COVID pandemic now on, you will know that they're talking in terms of a PCR diagnosis of COVID infection, a polymerase chain reaction, PCR diagnosis of COVID infection. Again, a molecular biology term. You need to understand what it is. In the pathology report that the pathologist sends back to you, almost every report nowadays, if it's detailed enough, consists of a few molecular biology terms, at least a few. And if you're not familiar with those molecular biology terms, you will find that, yes, you're going through it, but then you're skipping the parts that deal with molecular biology. Instead, if you know the basics of molecular biology, and especially its principles, its fascinating principles, you will realize that when you look at pathology reports, it'll make sense to you. Now, for a modern understanding of cancer, when they speak in terms of oncogenes, regulatory mechanisms, point mutations, deletion, addition mutations, if you don't understand molecular biology, you really cannot understand the modern understanding of cancer. The understanding of cancer that it actually is a metastatic phenomenon is from, in fact, the 19th and early 20th century. The molecular understanding of cancer is basic to all kinds of modern medicine, including surgery. Now, when you look at surgical research nowadays, even research that deals not necessarily in molecular biology, a lot of the research coming out does have molecular biology terms in them. It'll have things like oncogenes, point mutations, PCR, gene sequencing, and you should know what that means. Because even if you're not going to be doing full-time research or heavily involved in research, you will throughout your life being brought face to face with advances in surgery, and if you're going to be reading those papers, you should know something about molecular biology. And of course, if you want to impress your pathology, internal medicine, ID colleagues and your family, if you know molecular biology, of course, that is preferable. So, the reasons why a surgeon must know molecular biology also consists in some way of the objectives I hope to cover in this lecture. There are 4 essential areas in molecular biology, and this is something I think is a guiding basic principle in molecular biology. Because many people think molecular biology is just DNA, and that was true until probably 1975. And so when people think molecular biology, they think DNA, and that's fine because DNA is basic. But what we have realized in the last 50 years or so is that RNA and protein is just as important as DNA. So, as a matter of fact, why does DNA exist? It is to make protein. The reason for DNA to exist is to make protein. Next to body water, we are mostly protein in weight. And everything basic to life in the human being or in organisms is mediated, carried out by protein. Protein has structural functions, um, functions. Enzymes are proteins. So proteins is very basic to life. RNA is also very basic to life because DNA speaks, as it were, to RNA. And so you need to know that all these four areas are equally important. How does the cell interact to control the DNA, the RNA, the protein, the interaction with each other? That's the other fourth big area of molecular biology, all equally important. Now, you are probably familiar with Mendel. He was an Austrian monk. He did experiments in his back garden. Long plants and short plants, crossed them. Some were long, some were short. He looked at how they were crossed and deduced that there's a dominant way of transmission of these genes. He didn't know about genes at the time, but the way the crossing worked made him deduce something about transmission of hereditary characteristics. That was very basic to the idea of genetics and molecular biology. But it wasn't until 1919 that Levine discovered that DNA is composed of nucleotides. That's in capitals because it's a new term. And a nucleotide is nothing but a nitrogen base, and we'll come to what a base is afterwards, a nitrogen base combined with a sugar and a phosphate group. Sugar, phosphate plus nitrogen base is a nucleotide. So the nucleotide is inside and the phosphate sugar base is the backbone. The phosphate sugar base, in a sense, carries the nitrogen base. And you all know that the nitrogen bases are of two types. The purines adenine, A, and guanine, G, and the pyrimidines cytosine and thymine, T. Now, everybody knows Watson and Crick and the helical DNA model, but there was a very basic advance, which we now know as Chargaff's rule, which came out in 1950. The Chargaff's DNA rule simply states that the larger basis purines, A and G, always exist in a 1-to-1 ratio with the smaller bases called pyrimidines. So A and G is always in the same proportion to C and T, always in a 1-to-1 ratio, and that's regardless of species. So how is that possible? So Chargaff's rule was a puzzle asking for a solution. And that solution came in the momentous advance that occurred in Cambridge in 1953, when Watson and Crick unraveled the helical structure of DNA. Watson and Crick essentially solved the puzzle of Chargaff's rule. How come A plus G is always equal to T plus C, regardless of species? Now, they solved that puzzle, but in addition, what did they do? They proposed and confirmed the double helix structure of DNA. Watson and Crick put the bases on the inside of the double helix and the sugar phosphate backbone on the outside. And the two strands were linked by weak hydrogen bonds. So this is, I don't actually need to show you this because this is imprinted in everybody's mind nowadays. Even kids know the structure of DNA in a very general way. But you can see the yellow phosphate sugar backbone and the colored bases, the nitrogen bases, A and G and C and T, linked weakly by hydrogen bonds. Okay, so what was so brilliant about the double helix? What Watson and Crick realized and transmitted to the world is that the coiled structure meant that a lot of genetic information could be packaged super efficiently in the available space. But in addition to the brilliant packaging, the two coils could separate easily, each coil replicating itself. So not only was a lot of genetic information possible to be packaged into the DNA by this helical coil structure, you could also separate the two helical strands easily, each coil replicating itself. This gives rise to faithful duplication, each strand copying itself exactly by base pairing to give two identical DNA molecules. Right. Now, the public, when they understand or think of genes, they talk in terms always, or almost always, the layperson will talk in terms of genes as what a father transmits to his daughter, or a mother to his daughter, her daughter, mother to her son, that sort of thing. So those are important. Those are hereditary genes, but that's not what the molecular biologist thinks of as a gene. A gene for the molecular biologist, and therefore for the surgeon, is the DNA material required to code one protein. So a gene codes one protein. And you realize that I've put gene there in capitals too, because you can always go back to that and see what is the definition of a gene. What is the definition of a codon? A codon is made up of 3 nucleotides. Now, if you recall, a nucleotide is one base, phosphate, sugar, phosphate and sugar. One base, phosphate and sugar, 3 things together is called a nucleotide. And each codon is composed of 3 nucleotides, and each codon codes for 1 amino acid. So a gene codes for 1 protein, a codon codes for 1 amino acid. The gene and codon information in each organism is its genetic code. And there are 20 amino acids, as you know. Some are essential and some are non-essential. There is such a thing known as the central dogma of molecular biology. It was proposed by Francis Crick in 1958. And the central dogma of molecular biology is what the figure on the right illustrates. And that is that information flows in one direction only, from DNA to RNA to protein. In other words, DNA is transcribed into RNA. It's like somebody giving the old days, they used to have these runners from city to city who take messages that were transcribed into a scroll. So DNA transcribes messages to RNA, which then acts as a messenger, carrying the information to be translated into proteins. So the direction is DNA to RNA to protein. Now, since that time, we know that RNA interacts with DNA in a reverse direction, like in the retroviruses, for example, the HIV virus. Reverse transcriptase is the enzyme that makes that possible. But you've got to remember that Crick did not preclude the flow of information from RNA to DNA, but only said that protein cannot send information back to DNA. So the central dogma of molecular biology still holds, still stands, that DNA transmits information always to RNA, RNA to protein. Now, what are chromosomes? Now, again, chromosomes, because people learned this in school, the meiosis and mitosis and so on, they think of chromosomes always in terms of, your mother's chromosomes to your daughter, mother to son, which is the hereditary aspect of it. But essentially, for the molecular biologist, for the surgeon, the chromosome means something different. It's essentially the structure in the cell into which DNA is packaged tight. If the DNA of each cell was not packaged in efficiently, it would be several feet, literally several feet in length when unrolled. So DNA is packaged densely into chromosomes. And chromosomes therefore contain lots of packed DNA. And as you know, there are 46 chromosomes in every human somatic cell, 23 pairs. The other great advance in molecular biology had to wait another 50 years after Francis Crick and Watson. And the Human Genome Project unraveled the entire human genome in, actually it was completed in 2000, published in 2003. It was launched in 1990, so it took about 10 years. It was probably the biggest international collaboration in science ever. It involved a collaboration between 20 countries. It was led by the present director of the National Institutes of Health, Francis Collins. Some of you might have read his book, The Language of God, which was a New York Times bestseller when it came out. And if you've been watching some of White House COVID-related press briefings, there's a fellow called Tony Fauci who keeps coming up. Francis Collins actually is his boss. And Francis Collins' group identified all the genes in the human genome. It coincided with the 50th anniversary of Watson and Crick's discovery. What did the Human Genome Project show? It showed that human cells have approximately 20,000 genes that code for proteins. And these are contained in 3 billion base pairs. I think in Francis Collins' book, he says that if you were to read out the base pairs, it would take something like 30 years to read out all the base pairs. Now, what are exons? Again, remember the capitals mean that there's a new term coming. Exons are the transcription unit part of each gene that code for amino acids. So there may be a gene that is related to one protein. One gene, one protein. However, not the entire gene doesn't necessarily code for amino acids. The part that codes, only the part that codes, the transcription unit, is the exon. And the exons are separated by introns, which are non-coding units. Exons, introns. Now, each gene is one of a pair or allele of another gene. So if there's one gene that has a characteristic function, there's usually an allele, the corresponding gene, carried in the corresponding chromosome pair. The exception, of course, the XY chromosome pair in the male, as men have only one X chromosome. What did the Human Genome Project show about the function of genes? As you will see, not very much. We know the structure of genes, we know where they are, we know what they are. We don't know exactly how they do, and we know even less about how they interact with each other. We do know that less than 1.5% of the genome is made of exon sequences. 80% of the genome, remember, genome is the sum total of all the genes in the human body. 80% of that is estimated to be involved not only, because that's really, really important, estimated to be involved in regulatory functions. The rest are composed of repetitive sequences, which are also hugely important, it turns out. Now, a very few of the human genome code does code for RNA molecules that do not produce protein, but play a regulatory role in gene expression. The Human Genome Project showed us the structure of the human genes, of all the human genes. But as to the function of the human genes, we know less, and we know very much less, actually, and even less about how the genes interact with each other. But this was an enormous milestone in science. Now, some more new terms. What are telomeres? Telomere sequence is a repetitive sequence of DNA. The sequence is TTAGGG, found at the ends of a linear chromosome. Their role is to prevent chromosomes from shortening during DNA replication. Now, I don't know if you've heard about the research that is going on about the telomere of lobsters, which apparently don't shorten, or if they do, they repair themselves, and so they live for up to 300 years or something. So telomere loss, we know, is associated with aging and cancer. And then what is the histone? These are things that you keep reading about, hearing about in science. You keep reading about and hearing about in various forums, in research, and all these really important fields. Now, a histone is DNA with protein. That's all it means. In order to exist in a stable state in the cell, it has to be packaged with proteins. And the protein plus DNA is called a histone. And the chromatin is the combined DNA plus histones, highly ordered, like beads on a string. So that's what it's called. It's like a beads on a string structure. And that's the chromatin, which is then basically stuffed into a chromosome. I've shown that through the diagram on the right. You have the double helix, and then with the histones, the chromatin fiber is sort of a beads on string appearance that is then stuffed into the chromosome. Fascinating. There's also another form of DNA. As you know, most DNA exists in or inside a cell, but there is a type of DNA called mitochondrial DNA, or mtDNA. It's smaller in size than nuclear DNA, but this is the DNA that is used in crime scenes and in human identification. We know that it's passed consistently from mother to offspring across hundreds of generations. So if they find DNA in a particular area and then the same type of people in another area, they know that they originally were in one area. So for anthropology studies, mitochondrial DNA is quite important. Now, the DNA replication sequence, I don't have to tell you that the two strands go apart and then replicate. That's known to everyone. You know that the exposed strand acts as a template and then makes another strand of DNA, and so one DNA molecule will get two. Here's another very important term in capitals: DNA polymerase. It's the enzyme that makes DNA replication happen. Enzyme is a protein, as you know, but DNA polymerase is the enzyme that makes DNA replication happen. The RNA primer starts this reaction, but it's facilitated by DNA polymerase. Once replication is complete, RNA primer is removed. So RNA primer, DNA polymerase, and off you go. The DNA starts replicating. But once the DNA is finished replicating, the RNA primer is removed. That's another amazing control mechanism. The DNA then zips up, rewinds, coils, and replication is complete. Now, DNA replication is amazingly accurate. The error rate is only 1 in 10,000 to 1 in 1 million. Can you imagine the billions of pairs of bases that are replicating, but the error rate is only 1 in 10,000 to 1 in 1 million? Now, Partly and importantly, this is because the enzyme DNA polymerase carries an up and down proofreading function. This is like, in some of the movies you've seen soldiers line up and a sergeant goes down the line of soldiers, and if somebody is looking disheveled or not quite like he should be in a military parade, he's pulled out and somebody else takes his place. So the DNA polymerase goes up and down as replication is taking place. And if it detects a nucleotide error, it will excise the incorrect base and insert the correct base. Really, really fascinating. Mismatch repair, another new term. Mismatch repair is the repair of a replicating DNA strand by DNA polymerase proofreading. And there are other equally important methods, and we'll come to that later, probably in the next seminar. So, if proofreading and other repair mechanisms fail, mutations become permanent. Again, these are terms you will read about, hear about very often, and so you should know what they are. A point mutation is when a single nucleotide is substituted for another. A single nucleotide substituted for another. Substituted. An insertion or deletion mutation is when one or more nucleotides are added or deleted. So there are basically three types of DNA mutations: a point mutation, an insertion mutation, or a deletion mutation. The protein that is mutated with the mutated DNA codes then either loses its normal function or it acquires a new harmful function, and both can cause disease. Sometimes the phenotype, or what the genotype actually, so the genotype is basically what occurs intracellularly in the gene, at the gene level, and the phenotype is what is seen clinically. Sometimes mutations are silent in that no phenotypic changes occur. So I'll give you an example of a point mutation, which is also fascinating. Sickle cell anemia is caused by the point mutation of a gene that codes for beta globin. Beta globin, as you know, is part of hemoglobin. So at the 6th position of beta globin, the point mutation replaces the amino acid glutamic acid with the amino acid valine. So glutamic acid is replaced with valine, just one amino acid. The altered protein causes the sickle cells to clump together, distorting the RBC to a sickle shape, and thrombosis and end organ damage ensue. So on the left side of this diagram is the normal RBC. What happens at the genetic level is what I put in the middle. And as you can see, the normal protein has glutamic acid. The abnormal protein has valine, just one amino acid substituted for another, and it gives rise to sickle cells on the right side of that diagram. Now, if only one allele of the sickle cell gene is affected, a less severe form of sickle cell disease results. As you know, this has been seen in areas that have been or are afflicted with malaria, and it is thought to give an adaptive advantage to the population because of that sickle cell, partial sickle cell. Some more terms. Again, if you have understood what I said before, this will come quite naturally to you. SNPs, GWAS, and polymorphism. In human beings, DNA is identical in 99.9% of the population. No matter what the color of the eyes, mouth, skin, hair, 99.9% of the DNA is identical with every other person. It is 0.1% that accounts for the diversity. Of course, the 0.1% can still combine in different ways. Nuclear polymorphism, another new term, is a population-wide difference in DNA that occurs in that 1% that is diverse. SNPs, pronounced SNPs, is single nucleotide polymorphism, and that's the commonest kind. GWAS is GWAS is genome-wide associated studies, again made possible by the Human Genome Project. It compares whole genomes of populations, example those with or without a disease, to look for SNPs. An early success of GWAS in modern molecular biology is that we realize that a variation in the complement factor H gene causes age-related macular degeneration in the eye. Gene cloning is not the same as whole body cloning like in Dolly the sheep. In gene cloning, only smaller regions of DNA are copied. The first step involves the generation of recombinant DNA. This is, I put this in because this is used a lot in drug development. Recombinant DNA is used for research for drug development, for therapeutic safety, that sort of thing. So it's a two-step process, recombinant DNA, which is artificially rearranged set of nucleotides. And then these recombinant DNA molecules are instigated to clone to the DNA of desired quantity. It's also used a lot in agriculture, like in GMOs. So when studying DNA, one of the basic mechanisms that are used is electrophoresis. Now, it's often necessary to identify specific fragments of DNA in a mixture. Gel electrophoresis, which you see on the left side of the picture, is essentially a process where, based on size, DNA fragments are separated on a gel base. So if you put the DNA fragments on a gel base, put a current across that gel base, the smaller fragments move much faster and they leave the larger fragments behind. And then the gel material is stained with special dyes, and all of you are familiar with how an electrophoresis slide looks like. It looks like bands, and the bands reveal the position of the various fragments by size. This is another term you will come across from time to time, southern blotting. It was not discovered in the South. It's not also practiced exclusively in the South. It's in fact named for a fellow called Edwin Southern, who described it. He was an American biologist who described it in 1975. And southern blotting is where when the DNA is on the gel, even though they are separated by electrophoresis, it's a messy separation. So Edwin Southern described an elegant technique when the gel is blotted onto a very specialized thin membrane or paper. So the DNA thus gets concentrated and accessible for identification and analysis by DNA probes, which are DNA sequences complementary to those that are being studied. What is the polymerase chain reaction? At the start of the seminar, I pointed out the fact that this is used for the diagnosis of COVID infections, a lot of viral infections. It's also used in a whole lot of other things. It's used, for example, in forensic science. You take a small quantity of DNA, there might be a very tiny quantity, a quantity that is useless for analysis, were it not for the polymerase chain reaction. So the polymerase chain reaction is a powerful DNA amplification process. So the key word here is amplification by several times, by a process of exponential growth in quantity. It's a powerful amplification procedure that makes it possible to make large quantities of DNA from only small and sometimes infinitesimally small quantities of starter DNA. Now, PCR was developed by a fellow called Kary Mullis in the 1980s. Again, I think he was American too. And he won the Nobel Prize for this in 1993. So this is the other reason you should know molecular biology. A lot of the Nobel Prize winners in physiology and medicine nowadays are in fact molecular biologists. Rightly so, because that is the cutting edge of science now. PCR is widely used throughout the medical and scientific world. It's used in diagnosing infectious diseases, including COVID-19. It's used in diagnosing genetic disorders, establishing paternity in crime scenes, as you know, in DNA cloning, and so on. How is PCR done? And here there's a fascinating story. Again, everything in molecular biology I find really fascinating because it is, in many ways, you've heard about the universe being billions of stars, even trillions of typical systems like the solar system, all being held together by a very intricate and finely controlled set of physical laws and mathematical laws, everything working together. In the cell, within the cell, there is a similar system whose workings we're only now beginning to understand. Now, since DNA polymerase is the essential enzyme in DNA replication, you need a supply of this enzyme for PCR. So, in the lab, to kickstart the replication and amplification of DNA, you also need heat to be applied. Now, this is common, as you know, in a lot of enzymatic reactions. Enzymes work best when there's a little bit of heat applied. And to kickstart the reaction, you need heat. Now, there was a conundrum here that researchers couldn't overcome, and that is that DNA polymerases available naturally are all thermolabile. In other words, they were destroyed by heat. How do you solve this? Kary Mullis solved it. So Kary Mullis started using a DNA polymerase called TAGG polymerase. He isolated this from the bacterium Thermus aquaticus. Thermus aquaticus. And Thermus aquaticus was found in geothermal hot springs. TAGG polymerase could withstand heat to high temperatures, and PCR became possible in the lab. So initially, when Kary Mullis started this and in labs, they used to do it essentially with a kettle, with laborious stop-start cycles with a kettle. Now, of course, you have automatic thermal cycling machines that make PCR possible on a huge scale. You'll also hear the term RT-PCR and qPCR. These are all variations of the same theme, amplification of DNA using DNA polymerase. In RT-PCR, the enzyme reverse transcriptase. So let's say there is reverse transcriptase in a particular virus. That virus, reverse transcriptase, then generates the DNA. The DNA is then amplified, and then you are able to diagnose which virus it is. So you have the RNA starter material, the reverse transcriptase, and then what is called complementary DNA, or cDNA. And cDNA is then amplified, the same idea, amplified DNA. RT-PCR, and then qPCR, or quantitative PCR, is DNA amplified and quantified simultaneously. DNA sequencing identifies the precise order of A, C, G, and T in a DNA fragment. Now you can sequence individual genes, segments of a genome, or even the whole genome, the whole human genome. So the initial method of gene sequencing that the fellows of the Human Genome Project used, Francis Collins' group, was Sanger sequencing. It was the first method of sequencing used. It was named for a fellow called Frederick Sanger. He won the Nobel Prize in 1980. So what is the importance of DNA sequencing? With DNA sequencing, you can make entire genome maps of life forms. They can be built and compared. You can compare human, animal, microbes. And then you can find these hard-to-find genes like those of cystic fibrosis. Francis Collins was involved in a big way in the finding of the cystic fibrosis genes. As you know, they're trying now very hard to find gene-targeted therapy for cystic fibrosis, and it looks like they're not far away. Um, I'll come to that probably in another seminar. Now, impaired genes like those of diabetes and cancer can be identified, and you can also find out which genes are preserved between species, and therefore there's probably a functional importance to those genes. All this can be done by DNA sequencing. And the sequencing that is used nowadays is what is called next generation gene sequencing. Why next generation? Because it's next generation from Sanger sequencing. And next generation sequencing has largely replaced Sanger sequencing. And what it does, it leverages the power of computing combined with innovative, what happens with a very powerful computer, which is in the lab. Not much more powerful than what you use, but powerful enough to do this. The computer is combined with an innovate, so the DNA fragment goes in one direction, the DNA probe is going in the other direction, and it's read continuously in real time, and then it's analyzed. When next generation sequencing first became available, it used to take several weeks, then it was down to 3 weeks, and now the entire genome can be analyzed in 24 hours. And the costs are coming down. It's less than $1,000. And I was looking up Ancestry.com, which actually offers one through their doctor. So a doctor in the US can order your gene sequencing just like a prescription. And you can get that for, I believe, $200 or $300 now. Personal genomics like that can identify defective genes rapidly, like those of cystic fibrosis, sickle cell disease. Now, what it can also do is it can work backwards from a rare disease to a probable gene. So what they can do is if they found families, let's say, with a particular rare illness, they can do a whole genome sequencing. They can compare it with normal genes, normal genome sequencing. They can see, what is the differences. And if there's a difference they can identify as being associated with that disease, obviously that's of great use to succeeding generations of that family. Like the Headley family in the UK in 2015. I'm not going to go into what happened there, but it's fascinating. They essentially found a family that had this rare gene that passed on hypertension mediated by kidney disease. Now, unfortunately, the Human Genome Project and then the gene sequencing cannot identify single genes for cancer and diabetes in most cases. In some cases, they can. But the molecular biology almost always involves a combination of DNA, RNA, and protein abnormalities and dysfunctions. But we're getting more and more information on this too. And the bioinformatics is the coming together of molecular biology methods with computational systems. You have biologists, mathematicians, physicians, computer scientists all working together and creating DNA databases that are now routinely deposited into publicly available websites. You can look them up, GenBank or the EMBL. You also have equivalent, equally important RNA and protein databases of equal importance. And I'll come to that in a different seminar. I want to close out this seminar by quoting from Francis Collins' speech when he and Craig Venter, the other guy who was involved in the Human Genome Project, they were honored by President Clinton in 2000 in the White House. And Francis Collins, the leader of the Human Genome Project, said in his speech, quote, "It is a happy day for the world. It is a humbling day for me and awe-inspiring to realize that we have caught the first glimpse of our own instruction book, previously known only to God." Well, thank you very much. That was the first of my 3 or 2 seminar lectures on molecular biology. You can tell me whether you want one more or two more of these seminars by email or by um, text. Get in touch with me maybe in the next 2 days. Thank you very much. God bless. All the best.